who is Laura Berkowitz, a graduate student from the Clark Lab at the University of New Mexico. Laura was in my Neuromatch Academy pod, so I am delighted to be able to MC for her talk today. She has a recent second author paper on how prenatal alcohol exposure alters hippocampal representations, but today she's going to be talking about her main thesis work on head direction cells in an Alzheimer's disease rat model. So please take it away, Laura.
All right. Well, thank you so much, Emily. And thank you very much to the Neuromatch organizers for organizing this great conference. So today I'm going to talk to you about postsubicular head direction cells in the TgF344-AD rat model. So let's just go ahead and get right into it. So spatial navigation is one of the earliest deficits to arise in Alzheimer's disease. And while at first this might just manifest as making the wrong turn on your way to the coffee shop, over time it can develop into pretty severe deficits, which it can result in getting lost episodes. And so while we don't know the mechanism underlying these changes, we do know that Alzheimer's pathology is found throughout brain regions that are thought to support spatial navigation. And so particularly we know that here's some images of postmortem tissue showing pathology within the parahippocampal cortex. And we know that from animal models, neurons in the hippocampus, such as place cells, which fire at a given place, exhibit instability, and entorhinal grid cells also exhibit decreased gridness in models of Alzheimer's disease. We know very little about cells that support spatial navigation within the presubiculum or postsubiculum in animal models. And so the focus of this research is to understand how head direction cells, which are a prominent spatial cell type in the postsubiculum, are affected in the presence of Alzheimer's pathology. So here I have a healthy circuit where we have our pyramidal cells, which are putative head direction cells, and our triangles, and supportive GABAergic interneurons or circles. And so from in vitro work, as well as in vivo, we know that these oligomeric species, these amyloid beta oligomeric species, tend to correlate with dysfunction of GABAergic processes. And so with that, we predict that head direction cells in the postsubicular cortex should exhibit increased firing rates and also unstable directional tuning as a result of this GABAergic interneuron dysfunction. And so to test these hypotheses, we implanted animals aged 15 to 17 months where pathology is quite moderate in the postsubiculum. Rats underwent a fairly standard head direction cell paradigm. This is an overhead view of the maze area. So we have cylinders with one prominent cue card. The animal was placed in a flower pot in between sessions and was disoriented, preceding and following a rotated session to check for cue control. And so I just want to familiarize you with some of the plots that I'll be using in the talk. So here is a polar plot. It depicts the 360-degree horizontal azimuth, and this is a head direction cell here. And so this cell fires maximally at about 270 degrees. We can measure the width of that firing with this R length, as well as how much directional information with directional information content. And here I have a tuning curve, which is just a horizontal version. The peak rate is how we're going to be looking at firing rates, and the preferred direction is how we assess changes in tuning across different environmental conditions. So we first wanted to look at globally whether pyramidal or interneurons within the postsubiculum exhibited decreased or increased firing rates, given hyperexcitability found in other animal species of Alzheimer's disease. And so here I have classified out putative fast spiking interneurons as well as pyramidal cells in gray. And we found that within the TG animals, we did see a slight increase, so about a hertz, 1 hertz increase in firing rate, but no such difference within the fast spiking interneurons. And so next we wanted to see whether or not head direction cells also exhibited dysfunction with their firing rates and or the directional tuning aspects. And so head direction cells were classified using a linear nonlinear GLM and a shuffled R length. Here's some example cells here. So I have 6 head direction cells from my control group, and 6 head direction cells observed from my TG animals. And so when you first look at them, they look pretty good, pretty normal, but then we do see increased peak firing rate within the head direction cell from the transgenic groups, about a 3 Hertz difference, a very slight but significant increase in directional information content within the transgenic group, but there's no differences in stability, so how stable that preferred direction is within a trial, or how wide that preferred direction is, although there is a little bit of increased variability with my transgenics. So overall, directional integrity seems to be fairly normal, despite the Alzheimer's pathology. And so next we wanted to see whether or not these head direction cells were able to maintain their tuning between 2 similar environments. So here I have scatter plots showing the preferred direction within 2 conditions. So baseline versus standard, standard versus baseline, standard 2 versus baseline, and standard 1 versus standard 2. And so given that the cue card is in the same orientation within these conditions, you would expect that the preferred directions would be the same within each environment, and it would align along this diagonal. And so indeed, we do see that there is some variance the further in time you compare, such as with baseline and standard 2, but overall, there's no difference in preferred direction tuning across similar environments. And so next we wanted to compare whether or not cells from the transgenic group were able to rotate with that cue rotation. And so when we plot the same conditions, so preferred direction between the 2 environments, we see that there's a little bit more variance, and this variance is present within both controls and transgenics, although it's a little bit more severe with the transgenic population. And so we wanted to see what could be contributing to this variability. And so you can look at tuning over time. So here I have my polar plots for each session on the top panel, and you can plot their firing rate bend across time, which is depicted in these heat maps. So this is time across head direction. And you can also look at head angle in gray with the when a spike occurred along the angular azimuth in colored dots here. And so this is an example control cell, and we see that the tuning is relatively stable across conditions. We note that there's a shift when the cue shifted and it was able to return back. But in the majority of cells observed for my transgenic group, I noticed that the tuning tended to be a little bit more unstable across sessions. And it also, I had an increased incidence of cells that assumed this drift of their preferred direction following the cue rotation. And so because this drift is associated with error accumulation with angular and linear velocity, we wanted to identify whether or not cells within the postsubicular cortex exhibited modulation to velocity. And so indeed, we found that both controls and transgenics had quite a large population of cells that were speed modulated, specifically for linear velocity. And so I have here an example tuning curve where we have running speed and firing rate, and you can see that this cell increases its firing rate as a function of running speed. Here's a similar cell from my transgenic group. And so we wanted to see whether or not these fast spiking interneurons were different, and indeed we found a small difference in slope. And so firing rate for the firing rate modulation associated with velocity was increased in my transgenic group compared to controls, indicating that there may be some dysregulation associated with those fast spiking interneurons. And because these fast spiking interneurons may be important for integrating angular velocity signals into head direction cells, we also investigated this within our head direction cells. And here I have an example cell here, which was found to be also modulated by linear velocity, and we found a similar increase, small but similar increase in the extent of modulation of the head direction cells within the transgenic group. And so overall, what we found is that firing rates were indeed increased in head direction cells within the postsubicular cortex, but this but we saw no differences in the tuning width or tuning integrity or stability. We found some evidence, which I will explore in future research a bit further, but some evidence that head direction cells from TG animals tend to drift following a Q rotation, and also that head direction cells and fast spiking interneurons tend to have an increase in their modulation for linear velocity. And so with that, I want to thank my research team. Many thanks especially to Ryan Harvey for his help with the analyses and the graphics, as well as my undergraduate research assistants, Katie, James, Maria, Victoria, and of course my PI, Benjamin Clark. Thank you so much for your time, and I'll now take questions.
All right, thank you so much, Laura. While we wait for some questions to come in, we have a few minutes for questions. I had a very basic question to start, which is how long are your recording sessions, and have you tried going longer to try to kind of like after the Q rotation, for instance, just to pursue the extent of this drift and whether it gets more severe over longer sessions?
Yeah, so so these Alzheimer's rats are, I mean, they're fairly old, right? They're 15 months. And so the sessions can tend to be quite long. So each condition is about 20 minutes. And then, right, so for a 4 condition environment, it can be up to an hour if the rat is doing really well, but usually it can be an hour and a half and maybe 2 hours. So they're fairly long, but that's exactly what I was wanting to look into. So I'm actually having longer sessions and including sleep sessions to see whether or not there's drift during sleep, as well as drift within the same environment over longer periods of time.
Very cool. I have another question, which is kind of a bigger question, asking first if I would kind of expect that if there's going to be some difference in any sort of head direction coding, that you might expect to see differences in heading direction when they're doing some sort of goal-oriented or like homing task, that they might have, a similar latency, but maybe the direction that they start out facing at the beginning of their trajectory is suboptimal, or somehow, the gain is too high or something like that.
Right.
Have you guys tested anything like that?
Well, we have assessed these animals in a water task, and while we didn't specifically look at heading error to the goal location, we did find that the animals made less precise trajectories to the platform. So while they were able to kind of swim towards the general area, they were off to a certain extent, which made them kind of loop around. So kind of exactly as you described. So yeah, so that does seem that could be a problem.
Actually, relatedly, there is a talk that was just happening in another room that maybe is a piece of software that might be useful. There's a piece of open source software called Pathfinder.
Yeah, I'm familiar with it. It's a really,
okay, great.
It's a really cool software to use.
Yeah, I love the idea of water maze strategies and using that to kind of extract more fine-grained information. Like, maybe these models have the exact same escape latency, but if all you look at is escape latency, you miss the fact that the strategies they're actually employing are like compensatory.
Right, and that's what we found. And I think the water maze strategies are a really good addition to any kind of water task.
I agree. We can wait a little bit longer for a few more questions. We have a minute or so left, but I had one more question while we wait, which is kind of, again, big picture. So you were talking about how this drift might lead to some sort of error accumulation. How do you think that might affect coding in downstream circuits like in medial entorhinal? Like would you expect, for instance, like we know, for instance, you can have error accumulation with path integration, where if you encounter a border, you kind of like error correct. Would you expect that there is any way for these animals to be able to error correct, or is the drift kind of infinite? And then how would that then translate to, you know, for instance, grids having representations that are also drifting?
Well, from the cell recordings and other animal models of AD, there is evidence that grid representations tend to become unstable and under amyloid pathology. And I mean, I think it's just in terms of what could correct for this drift, I think the alternative is visual inputs, and whether or not visual inputs or other types of sensory information is integrated into the circuit in an efficient way, or if it's also contributing to this drift. It's unknown as of now, but it's a good point of research to look into.
Yeah, I'd be curious to know, especially to figure out what whether there are upstream circuits that are involved in this potential drift. I know that there's some evidence that there are vestibular deficits in Alzheimer's disease models, or rather in models and in patients, that there tends to be some vestibular deficits that actually come before memory deficits in Alzheimer's patients.
Right, and that would be a fairly severe consequence to the head direction cell circuit, since it's pretty much dependent on it.
Yeah, quite heavily. All right, thank you so much. We are going to move